Yes. Now, let's begin with the topic today. It was just to catch your interest and know about what are the highlights of the topic, right? I hope you have observed the video and have recorded the the points. Have you? Yes? I don't get your answer, please. Please type yes or no. Is it audible to you? Hello? Hello? Yes, fine. So let's continue. Fine. Here we have seen one video, right? That is about a shotgun sequencing tool, the machine that is on the market. It is referred to as, of course, it is for sequencing and then analysis. So hi-fi, that was uh, the the name of the instrument, right? So here uh, we have to know the basic terminologies now, right? So when I say this, can you see this next slide? Right. It is the next terminology. What is it? It is saying that it is quantic. We have been talking about different things, and now the new term, that is quantic. Probably you have come across it while reading about, but to say in 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 short, to say, it is about um, the sequence, ATGC sequence, which is continuous and without gaps, right? So if this is the scaffold, then part of it here you see a gap. That gap is extracted and the ends are joined. So simple definition is that you are going to have a flow of ATGC sequence without gap. That is referred to as quantic. Now let's know about quantic in details little more. Contigs are made up of DNA fragments, and how the fragments are, there are no gaps. So you have to have overlapping fragments, right? They are made up of ATGC basis acid, and contings are created by aligning the overlapping fragments, and so that you can get a continuous long sequence, right? Now when I say this, what are the types of contings? Clone contings, or there can be sequence contings. Clone contings are those which provide a physical map of set of clone fragments. You are aware what are cloning, what are clone fragments? Of course, when we say clone fragments, it is about the libraries that are created, right? So when I say cloned contig, that means the fragments are cloned into some vector, you can say, and then then it is going to give you entire plethora of or entire sequence, long sequence. Or another type is sequence contig, where you have provide the DNA sequence of a genomic region. So you have not created that clone of it, right? So what are the steps in contig assembly? These are the terminologies which you will have to correlate when we are going to learn about shotgun sequencing. So fragments are generated out of the DNA, many copies are generated, then each fragment is cloned into a vector, sequence of each fragment from both the ends is done, and then you are going to align these sequences, compare the sequences, and find out the entire sequence for the particular region of the DNA or maybe entire genome. That is what is about clone counting, right? But you can have sequence contigs where you are not doing any cloning also, right? Now let's go to the next term, that is about shotgun sequencing. That is the topic for today. As the design in the slide, the schematic presentation you are seeing, you have a template DNA. Then you are going to go for shotgun sequencing, DNA sequencing. These are the titles I'm just reading through, right? Then analysis, and then you are going to assemble the entire genome sequence. That is how it is done, that we have to learn. Now, these are a few of the points in the history that it was first proposed in 1979 by Stadin to speed up the sequencing processes. We know the Maxwell Gilbert method and Sanger's method, their speed, their efficiency, we have talked about it. And right from Sanger's method to the NGS methods we have talked. So NGS matlab you have to talk about shotgun sequencing, right? So let's go into the details of it. Now, hierarchical shotgun sequencing and whole genome sequencing, these are the two types of it. When I say hierarchical shotgun sequencing, which is again the same terminology you can apply for it, that is clone by clone sequencing. And when I'm saying whole genome shotgun sequencing, direct sequencing of the entire genome without the initial mapping step or without putting it into clones, right? So when you are talking about the whole genome shotgun sequencing, here you are going to utilize the entire DNA, but how? Such a big long DNA cannot be handled at a given point, right? So you need to fragment it. So random fragmentation is done, and then it is sequenced. Now, why do you have to go for complete genome sequencing? Why not the earlier methods, which are say amplicon based methods, are enough? Amplicon based methods may say the most important one is 16S rRNA sequencing, which we all know in case of bacteria, it is going to be very important because the regions within the 16S rRNA sequence are the most conserved sequences, right? So you are going to make use of these preserve conserve sequences to find out if any variations are there, any alterations are there, any addition mutation addition deletion type of changes are there in the sequence, and maybe then you can assign it to the different genus, species, taxa, subspecies, subvar, etc. We all know that when it is more than 95% of similarity in the DNA sequence with the known sequence, when you compare it with, you call it as of the same genus. And when there is 99% similarity, we say the organisms they belong to the same species. Of course, these are the accepted terms. However, they also have certain limitations. Okay, now why to go for shotgun sequencing when you are talking about the method like 16S sequence, 16S rRNA sequencing, right? So phylogenetically and taxonomically very informative markers you have in 16S rRNA, for example, right? In fungi, you have ITS regions in their 16 ki jagah 18S rRNA. We know that, right? So this, if such informative markers are available, then what is the need for 60 shotgun sequencing method? That is a question, right? We all know that eukaryotes, prokaryotes, all these communities, uh, and their cataloging is done with the help of these sequencing methods. But the concern is you have many organisms in the system, and how do they interact? Metagenomic approach, when it comes into the picture, then 16S rRNA alone is not going to be enough, right? 16S rRNA locus, which is taxonomically and phylogenetically important marker, I understand, but at the same time, how do these microbes they interact with each other, and what is the role of fundamental role of each organism in a given community is to be understood. Then small markers like 16S rRNA sequencing alone cannot be of use. You will have to know the entire gene sequence of that organism, what genes are functioning, what kind of role those microbes will have. Is there any similarity, dissimilarity from phylogenetic perspective? Yes, you have to find out. But in addition to that, when it is to metagenomics, right? So functionality, the role, the fundamental metabolic processes of the organism in a given niche are to be understood, then such only small sequences like 16S rRNA sequence or rDNA sequence, if it is used, it is going to have limitations. That's why you have to go for complete genome sequencing, right? Now here this slide shows you the amplicon sequence studies, which are based on 16S rRNA locus. Of course, they are informative markers, and a tremendous amount of microbial diversity on the earth has been what you can say discovered using these methods. Right. So you can see in the picture, hot springs are there, Arabidopsis thaliana roots are there, volcano eruption sites are there, or sites, or ocean thermal vents. That is what we call. They are there. So when you are going to have different microbes which may be common in all of these, is it possible? Are there any sequences which could be similar in the bacteria which are isolated from such diverse environments? Isn't it very interesting to know about it, right? And that cannot be understood unless you sequence the entire genome of the organism. Sequencing the entire whole genome by simple methods like Sanger's and all, we have talked the limitations of. The NGS sequencing, yes, that is what is different types of NGS. They where you call it as shotgun. The word itself is telling you, right? It's like a entire plethora of the functionalities in terms of genes and gene sequences. They are described, right? So how this shotgun sequencing is going to work, and what kind of methodology it is that we have to learn. So when we talk about amplicon sequencing, it has got limitations, as I said. What are the limitations? Let's know in details. One, I said ki uh, uh, how you are going to compare organisms if they are similar type or same type of group of organisms are present in the rhizosphere soil and that in the hot water springs. How you can go for that is one. You have to sequence the entire genome. But when we say about amplicon based methods, surely amplicon amplification, so PCR, so limitations of PCR are applicable here also, right? So there are biases and limitations which are associated with PCR. So um, you cannot resolve the substantial fraction of the diversity. सारी community आप उसके साथ, I mean with the help of PCR amplicons, you cannot reveal, right? That is one. Second is amplicon sequencing can produce widely varying estimates of diversity. That is another problem, right? Amplicon based मतलब marker gene है, उसका primer डाला, उसको amplify किया, then you sequence and try to blast the sequence and find the phylogenetic position of the organism, right? So amplicon sequencing can produce uh, wide estimates of the diversity. So you may not be 100% relying on it, right? And it is not me saying, people are talking about it since they started handling the big datas like say for example human microbiome project. All right. So while handling these, of course there are such a diverse um, human community also, if you talk about, and the estimate of diversity is going to be not 100% or very wide variations you will get if you talk about amplicon sequencing methods. Different genomic loci have differential power at resolving taxa. So एक एक sequence जो है उसका to resolve a taxa, right? Based on a particular small sequence, you are trying to resolve the taxa. So it is going to be different for different genomic loci. Certain loci, 16S RNA is one of them, but it cannot be 100%. So each one has to be explored and utilized. In addition, when you talk about amplicon-based sequencing errors and incorrect assembly of amplicons. You have heard about chimeras, right? So chimera generation could be one of the problems. It can produce artificial sequences by a random assembly of amplicons. And so such artificial sequences will lead to a chaos, a difficulty in identification of the organism and its phylogenetic position, right? Further, amplicon sequencing can provide an idea, you can say that, about the taxonomic position of a particular bacteria in a microbial community, but it is really impossible to resolve biological function associated with the taxa. right? So that is the limitation of amplicon based method. In some cases, phylogenetic reconstruction can be used to find out the biological functions that are encoded in a genome containing particular 16S sequencing. उसमें भी कुछ functions हैं, they can be used, but it is not complete picture, it is not complete scene, right? So it is going to have its limitations. So for accuracy, the methods that can estimate, you can say, the true functional diversity of a community, that is required. So true functional diversity of a community. So how is the genomic diversity of the community is there? How well it is represented by the genome based on the available sequence database? All these are going to be the challenges, but you have to make it more and more rich. The complete sequence databases are going to give you a better picture of genomic diversity. So amplicon sequencing, when you say, it is limited to analysis of taxa for which taxonomical information is available. But when we talk about uncultured organism, right, you don't have a database, then how you are going to go with, right? There where you need to understand the diverse roles of the organism, the functionality of the organism in the given community, and that is you are going to blend of these, and you call it as metagenomic approach. That's why you need NGS system, next generation sequencing systems like Illumina and Nanopore, and many more we talked about, right, if you remember. So you have to know ki, the entire genome sequence. If you want to know, then how you are going to approach it. So shotgun metagenomic sequencing is an alternative approach when you want to study the entire community, when you want to study the association between host and the bacteria, or a community and diverse microorganism in a community, or when you want to study the uncultured microbiota, then you have to go for shotgun metagenomic sequencing. All right, I'm sharing another slide. I hope parallelly you are taking the key points. I can share these what we say video again with you, but please try to take the key points so that you can understand and correlate while listening to it again. Right, now when I say this, can you see what you see in this picture, right? A natural environment, and the sample is being collected, right? So you want to find out who are those bacteria and what are they capable of doing. So capable of doing is their functionalities, and what are they is their phylogenetic position. So that is what is metagenomics approach by shotgun sequencing you have to talk about. So taxonomy and functionality, both you are putting together. So environmental sequencing approach, that is one. Second is non-targeted sequencing. When I say about shotgun non-targeted sequencing, what does it mean? When I say 16S rRNA or PCR based method or amplicon based method, I have a primer, right? I know a specific sequence, and based on that I'm trying to amplify it. But here I have the target, but in case of shotgun, I don't have any target. So it is referred to as non-targeted sequencing. Uncultured components, yes, because bacteria are not culturable. How will you identify their DNA? How will you isolate their DNA? How will you sequence that DNA? So you have hardly any idea about their those bacteria. So you are going to take help of shotgun sequencing in that. And of course, host associated microbial communities, then in diseases or in probiotics, or maybe you can say spread of a particular species in an environment, the antibiotic resistance, so many things you can explore using this methodology. Right, now let's look into the flow, how it works. So when it comes to metagenomic sequencing, you have to see two things, that is who is there and what are they doing. All right, so when I'm saying who is there and what are they doing, you can see in the slide that taxonomic diversity you want to find out. So marker gene analysis, that is what amplicon based, right? Then second is taxonomic diversity, phylogenetic diversity, to find out the novel taxa which is based on binding. right? So what is binding? The grouping sequences into defined taxonomic groups. Here is the definition for binding. Point number two. So, analyzing taxonomical informative markers. Second is grouping sequence into defined taxonomic groups, that is referred to as binding. So, you are creating the groups, you are putting them in specific groups, right? And then you are assembling the sequence into distinct genomes. That is the flow chart, you can say. In case of metagenomics, taxonomical diversity is quantified, and you are going to have these three things: analyzing taxonomically informative marker gene, कौन सा है वो specific marker gene that will tell us about, right, the functionality model, then grouping sequences into defined taxonomic groups, and then assembling sequences into distinct genomes. Of course, not at a time only single one you are going to handle, or there is no specific sequence. At a time, you will have to run these, all the three approaches you will have to explore. Maybe you are going to group the sequences first and then go for analyzing the taxonomically where they are related. Right. So when I say marker gene, for example, let's take very simple example. If I say rhizosphere bacteria like Rhizobium, the marker gene would be nitrogenase producing enzyme gene, isn't it? So such marker gene you have to find out in a specific community, depending on which location is there, what kind of molecules are present in that environment, what kind of system it is it living organism, what kind of organism it is, and so on and so forth, right? But is we have to just see the overview of the methodology. In details, it is going to take years to learn and masterize it. Okay, okay. We are talking about what are the steps. So, grouping the sequence into defined taxonomic groups, analyzing the taxonomic position of the marker gene, and then assembling sequences into distinct genomes. So you are going to have n number of bacteria in that sample. All the DNA is extracted. It is been sequenced. You have to categorize it, right? So that categorizing into taxonomic groups is binding. Is it okay? Right. And then you have to go for assembly and then analysis. And second is you have to check for the genetic diversity, which maybe predict what kind of functions they are doing. Are there any novel genes present that you are going to extract? And another thing is functional annotation, binding, all these are, or contig, these are terms which are associated with software systems, or you can say computerized systems. I should be very precise. Computing data jab aata hai, that time you get these terminologies. So functional annotations you can find out with reference to specific protein family if they have, or specific fatty acid profile they have, like that. So when you are going to have metagenomic sequencing, you have to compare all these things, right? You have to intercommunity similarities you have to find out, intercommunity variations you have to find out. You are going to have the metadata, the correlations you have to extract, biomarker based detections you have to do, so on and so forth, right? So this is the overview of how it works. Now let's go into little more detail. When I say taxonomic classification, marker gene analysis, right? So marker gene, as I said, nitrogenase enzyme, the niche gene, and how many bacteria do they have it? Is it present in the bacteria in only which are associated with plants, or is it going to be present in say Himalayan region soil also? That I want to check. For example, so marker gene is that nitrogenase enzyme, right? So marker gene database you are going to create, you are going to sequence that, you are going to compare that, and then you are going to generate taxonomic classification, that is the phylogenetic tree you are going to construct, right? Second is binding, as I said earlier. So in binding you are going to group into different groups, and then you are going to go for finding the sequence. Again, the steps are going to be similar. You have to go for marker gene database, इसका सीक्वेंस आपको निकालना पड़ेगा, डेटाबेस क्रिएट करना पड़ेगा, देन यू विल हैव टू कंपेयर दैट, एंड देन यू विल गेट द phylogenetic tree construction that you have to do, right? So maybe initially you are going with composition classification, or you are going with clustering. So Then second approach is sequence database, then compare it and then construct the tree. Third is genome sequence, database comparison, and then fragment recruitment, and then go for it. Ultimately, you are going to talk about phylogenetic abundance profile only, right? But with reference to taxon or total, that is only going to be different depending upon the approach that you have taken. And genome or contig coverage, so a particular sequence that you are looking for, right? So this is what is contig assembly. So when I'm saying contigs, these are small fragments, right? So these fragments you are going to join together, and then you are going to have a complete sequence generated. ये क्यों लगेगा, कैसे जनरेट होगा? द वेरी फर्स्ट पॉइंट दैट वी सेड इज एंटायर जीनोम यू आर गोइंग टू प्रोफाइल, and for that, at a time you cannot do that. Yes, now with NGS systems like Illumina, entire genome can be sequenced, but the point is, there again, are you going to go by random fragmentation method, or it is going to be an entire genome put into a small nanopores and it is analyzed? Yes, these days it is possible that entire genome you are going to get sequenced in one go, and multiple sequences. We have learned it in nanopore, but here the concept is the one which had arrived before nanopore technology. NGS का सबसे advanced nanopore methodology है, right? So coming back to the point, here you are going to have the sequence fragments, maybe marker gene based or maybe random fragments, and then you are going to generate the contigs, and then contigs contigs are going to be uh, arranged, and then you are going to analyze those, right?